Okay, and you are now the host. Hopefully you received the baton.
Okay, let me see if I can. Okay, can you guys see this?
Yeah, so let me introduce you. We'll just wait another minute or so, Bob.
Okay, you want me to unshare?
No, that's fine. I can see it. If you want to just go to your title slide, or is this your title slide?
That is my title slide.
Okay, perfect.
Thank you.
And you just have to put it in the larger view. Right now, I can see your list of slides on the left. That's perfect. Thank you. So welcome, everyone. We're just going to wait a few more seconds. I see quite a few people rolling in. Thank you for coming. I do ask you to turn your microphones off. Depending on how long Dr. Speth's talk is today, we'll have some time for questions at the end. If not, you can watch his recording. So this is being recorded today. I do hear a little background noise, so if you don't mind turning your microphone off, that'd be great. Okay, well, I have that it's 12:01 Eastern time. I'd like to begin. So good morning, good afternoon, good evening to everyone. Thank you for joining our Dementia Journal Club. I'm your host, Benedict Albensi, and we're broadcasting live today from Nova Southeastern University at the College of Pharmacy in Fort Lauderdale, Florida. And really, it's my pleasure to introduce our speaker, Dr. Robert Speth, who's a professor here in the Department of Pharmaceutical Sciences at NSU. And I just met Bob just a few months ago. So it's been great getting to know him and learning about him and his research. So before coming to NSU in 2009, Dr. Speth was the department chairman for the pharmacology department at the University of Mississippi, also known as Ole Miss. And he's also held other positions over the years, including one at the Cleveland Clinic in Ohio and at Washington State University. He obtained his PhD in pharmacology at Vanderbilt University, and that was under the supervision of Dr. Hank Yamamura, if I have that right. I hope so.
That was my postdoc.
Oh, okay. I'm sorry. Okay. Oh, that's right. So PhD in pharmacology at Vanderbilt, and then under the supervision of Dr. Yamamura, at the University of Arizona in Tucson, you did your postdoctoral work. I apologize. So Dr. Speth has several awards and distinctions to his credit, including establishing the Peptide Radio Iodination Service Center at Washington State. I think I pronounced that correctly. He's also a founding officer of the Society for Veterinary Medical Ethics and the founding editor and past president of the newsletter of the Society for Veterinary and Medical Ethics. He also received the Lewis Cattell Memorial Award in 1998 for his efforts in advocating for biomedical research in animals. And in addition, Dr. Speth received the Alumni Award from Tuskegee University, the Washington State Library Award, and a Washington State University Science Library Award for his advocacy and service on behalf of the Washington State University Libraries. So quite a few distinctions there for Dr. Speth. In 1995, he was elected a fellow of the American Association for the Advancement of Science. And to date, he's published over 200 scholarly works. So a lot of his work, as I understand it, is focused on the regulation of cardiovascular system by the renin-angiotensin system with a particular focus on the brain. And one of his papers was really the first report of the benzodiazepine binding site in the human brain. And he also had the first report of the distribution of angiotensin receptor subtypes in the rodent brain. So I do hear some background noise. If you don't mind turning your mic off. But today, of course, he's here to talk about the Christchurch mutation and other genetic modifiers of Alzheimer's disease risk. So welcome, Dr. Speth. Thank you for joining us today.
Thank you very much, Ben. Yeah, I would like to say, I would like to dedicate this to the memory of Hank Yamamura, my postdoctoral mentor, who is the first person ever to make me believe that I could actually be Dr. Speth. And he was one of the best mentors ever. And even though he's passed on, he still continues to be the inspiration for me. And I would not be the scientist I am today without his terrific mentorship. So today, my original plan was to do 3 papers. The first one, this Christchurch mutation, which I've been fascinated with for a couple of years now. But in the process of, re-investigating this paper, I realized that tau needs to get a lot more notice. There's way too much notice being given to amyloid beta, and tau has sort of been considered to be secondary. And I think, I had an epiphany while I was putting this talk together. And it's like, no, I think we need to think of tau before we think of A beta, amyloid beta. So I'm going to talk a little bit about that. And that is a little bit of a detour. And I don't know whether I'm going to be able to get to the MEF2 resilience to Alzheimer's disease or the AIB3 mutation as a contributor to Alzheimer's disease. But we'll see how it goes. Ben, stop me when the time is up, because I can talk forever. So anyway, let's get started. I first learned about this from a New York Times article by one of my favorite writers, Pam Bellick, who has actually come to do a presentation on scientific writing to our Georgetown Biomedical Scholars Group. So this paper, which was it's a Nature paper, a Nature Medicine paper, came out a little over 2 years ago, and it's talking about a woman who's part of the PISA cohort. Some of you may have heard about the PISA cohort. It's a large cohort of individuals, perhaps up to 1,000 people, that have the presenilin-1 gene mutation that causes them to have early onset Alzheimer's disease. And the person who discovered it and has been working with this group for several decades now is Francisco Lopera, who is one of the most heroic people that I know of for going out by himself through the outskirts, the rural areas of Colombia, to find these people and to study this cohort of people with early onset Alzheimer's disease. So basically, this woman had the presenilin-1 gene mutation, which by all means meant that she should have had early onset Alzheimer's in her 40s, and she should have died before she was 60. But remarkably, she had no cognitive decline until she was in her 70s. Okay, so this paper said that, hey, she's got another mutation. So this is the Nature Medicine paper that came out just almost exactly 2 years ago now, showing that she had an incredibly rare mutation called the Christchurch mutation in her ApoE3 gene. And what was even more incredible and almost astronomically remote is the fact that she was homozygous for this Christchurch mutation in her ApoE3. So just a little backup. Presenilin-1 is a mutation in the presenilin-1 gene, which is the catalytic subunit of gamma secretase, which is one of the enzymes that is metabolizing amyloid precursor protein. And this mutation in the PISA cohort is actually a glutamate substituted in position 280 by an alanine. And again, it causes early onset, 40 to 50 years of age, Alzheimer's disease. It is an autosomal dominant with 100% penetrance. However, the age of onset does vary as a function of other genetic characteristics. And one of the major genetic characteristics is the different ApoE alleles. So ApoE2 carriers have a much later onset of Alzheimer's than do ApoE2 E4 carriers, which have a much earlier onset. And we've heard a lot about ApoE4 as being an increased risk factor for Alzheimer's disease. So in this case, it hastens the onset of Alzheimer's disease in this early onset mutation Alzheimer's disease. So it's associated with overproduction of A-beta 42, which is thought to be the more toxic fragment of the amyloid precursor protein, leading to the formation of the beta amyloid aggregate plaques. So a little bit about the Christchurch mutation. It was discovered in the ApoE lipoprotein in a type 3 familial hyperlipoproteinemic patient back in 1987. It was shown to be an arginine substitution by serine in position 136 in the protein. And the low density lipoprotein binding domain of ApoE is approximately 130 to 143 amino acids. So this was right in the middle of the lipoprotein LDL, low density lipoprotein receptor binding domain. And it's considered to contribute to the hyperlipidemia that this individual was suffering. And basically, it was not binding well to the LDL receptor, and therefore clearance of lipids from the bloodstream was diminished. So these are some PET scans of the brains of the woman with the Christchurch mutation, as well as the brains of a person with the normal early onset Alzheimer's disease. So they stained amyloid with the Pittsburgh compound B. They stained tau with the F18 fluorotau-Sapir. And one of the things that's very noticeable is the fact that her brain was loaded with amyloid. Okay, the red is intense amyloid distribution. This is a medial vision, and this is the lateral vision of her brain. Okay, however, if we look at tau burden, she didn't have very much of a tau burden. In particular, I want to focus on this precuneus posterior cingulate region of the brain, which is thought to be the area of the brain where the tau accumulation causes the symptomatology, the phenotype of Alzheimer's disease. So then we compare with a typical PISA individual who has early onset Alzheimer's, and this is around the age of 40 to 50. So you can see there's some amyloid buildup, but not huge. But then if you look at tau, there's a huge buildup of tau in this brain, in particular in the precuneus posterior cingulate region. So what we have is high amyloid, low tau, and relatively cognitively normal. And then they also did some 2-deoxyglucose with a F18 label, and showed that her glucose distribution, or glucose utilization, excuse me, in her precuneus posterior cingulate region was pretty good, in contrast to the person with the early onset Alzheimer's, which had very, very low glucose utilization in this brain region, which suggests that this brain region wasn't functioning very well. So then just an aside here, there was a paper that came out not too long ago saying that, you know, basically the posterior cingulate and precuneus regions have deficient metabolic rates of glucose. and especially in ApoE carriers. And this shows up decades before the onset of measurable cognitive deficits.
Between 10 to 1.
So anyway, this
Could be already over. Might not even go home.
mutation is something that obviously is a potential lead for trying to find a cure for Alzheimer's disease. And we know that there's been more than 200 drug trials that have failed. Okay. And this is a little bit old because
We will call or text before they come.
it's more
They
than
haven't done that yet.
15 years.
It's either between 10 and 12, or late.
Sorry, Bob, I'm going to just tell everyone, I'm going to interrupt. Please turn your microphones off. We have some background noise that's distracting. If you wouldn't mind, please. I still see about 5 or 6 microphones that are turned on. If you wouldn't mind turning them off right now. Thank you.
Okay, thanks, Ben. So anyway, this seems to be a really good lead for what we might want to be looking for to start looking for therapeutics for treatment of Alzheimer's disease. And again, more than 15 years, well, actually, you know, the last treatment for Alzheimer's came out about 6 months ago, and it's incredibly controversial. And there's a lot of us who feel that it never should have been approved by the FDA, but that's another seminar. So what they found was that she really hadn't developed any very little Alzheimer's phenotype. She had mild cognitive impairment, but she was in her 70s. So, you know, it was very strange to them. So one of the groups that is studying the PISA cohort is the Banner Alzheimer's Institute in Phoenix. And Eric Ryman is leading that group. And he was a co-author on this paper. And that's when they found that she had this extremely, extremely, extremely rare mutation of the Christchurch mutation on her ApoE3. So again, ApoE4, we see the increase in risk, and it's present in 40% of the people with Alzheimer's. So this is just more details that are pretty much gone over already. Christchurch mutation was discovered in Christchurch, New Zealand, hence the name. So anyway, with the Christchurch mutation, she didn't show any binding to the heparin column that they set up. And let me just move forward on that. So again, you know, the promise of gene therapy possibly targeting ApoE to try and mimic what this ApoE3 Christchurch mutation is doing. So again, the huge amount of amyloid with very little tau suggests there's a definite dissociation of amyloid accumulation from tau pathology in terms of the cognitive decline. So I've always been kind of a Taoist, but I'm really a Taoist now, and I'm gonna give you even a little bit more Taoism as we move through this. So again, ApoE, we have ApoE2, ApoE3, and ApoE4, which actually don't differ a whole lot. But anyway, the ApoE2 has a lower risk, ApoE4 has the higher risk, and ApoE3 is thought to be neutral. And again, one of the interesting things, though, is that her ApoE3 Christchurch mutation again resembles this, causes this hyperlipoproteinemia type 3. So she had a history of dyslipidemia. So she wasn't clearing her lipids very well. She was on atorvastatin and an HMGA-CoA reductase inhibitor to lower her cholesterol. And they actually increased her dose after diagnosing and doubled her dose and added ezetimibe to help her clear the cholesterol. So again, the R136 is in the part known to bind to the LDL receptors and also to heparan sulfate proteoglycans. So compared to ApoE3, both ApoE2 and the ApoE3 Christchurch mutation have a 98 and a 60% reduction.
What he's talking about is all our researchers are dealing with.
Hence the,
and I have no idea what they're even talking about. All the enzymes,
increased
the
hyperlipidemia.
the whole part of science.
Anyway, HSPGs have been suggested as a way to promote amyloid beta aggregation and normal uptake of extracellular tau.
No, I'm not saying that. I'm just saying that it needs to have basic information just to be able to follow.
I wonder if the person that has their microphone on might be able to turn off their microphone. Thank you. Okay, so they're looking at the possibility that ApoE binding could be necessary for some of these effects. So this is an experiment they did. I think this is just fantastic. They took a heparin affinity column and poured over it the ApoE3 Christchurch mutation protein, ApoE4, 3, and 2. And as you can see, starting over here from the right, the black squares, the ApoE4 hung on to that column for a long time. It took a lot of the sodium chloride to elute the ApoE4 off of this heparin column. ApoE3 came off a little faster. ApoE2 hung on, well, did not have a particularly high affinity and came off fairly quickly. But then you look at the ApoE3 Christchurch mutation, it came flying off the column. It didn't take very much sodium chloride to elute the ApoE3 Christchurch mutation ApoE protein from the column. So again, this verified the fact that her ApoE3 had very, very little affinity to the heparin column, which was a surrogate for the LDL receptor binding. And then here we have, they made an antibody to the ApoE epitope defined by the amino acids 130 to 143, again, the putative binding domain of the ApoE4 for the LDL receptor. And sure enough, when they incubated the ApoE3 with the antibody that neutralized the receptor binding domain of ApoE, it came flying off the column. Now, they didn't have 100% occupancy of the ApoE, so some of the ApoE3 still eluted at the same place where the control ApoE3 eluted. Again, you had to have a fairly high sodium chloride gradient to elute it compared to the lack of affinity of the ApoE3 that had the receptor binding domain inactivated. So again, very compelling documentation here. And again, this is suggesting that, hey, if we can somehow recreate a monoclonal antibody, I know monoclonal antibodies aren't particularly popular for treating Alzheimer's disease right now, but if we could make the monoclonal antibody against the ApoE, ApoE3 or particularly ApoE4, we might be able to decrease the development of Alzheimer's disease by somehow preventing the tau accumulation. So last year, again, just about a year ago, this woman died, and she donated her brain to science. She was 78 when she died, and it was only at 72 where she just started to get MCI, and she wasn't demented at 78 when she died. So she has contributed her brain, but interestingly, she had a very low plasma neurofilament light chain, a blood marker for axonal injury and neurodegeneration. that was also an indication of the resistance of the ApoE3 Christchurch mutation for preventing tau pathology and the neurodegeneration, despite this high beta amyloid plaque burden. And I just threw this in because I was using this as a student presentation just to give the students an idea of the history of Alzheimer's disease. It dates back now 115 years. And this was the first woman who was diagnosed with Alzheimer's disease. So following up, her brain is going to be studied by a number of people. And this New York Times article was just talking about this investigator, Ken Kosick in Santa Barbara, who is going to be looking at the chemical receptor associated with the spread of tau from cell to cell. And I'll talk a little bit about that when I talk about Brock staging. And it's again thought that the ApoE protein may be involved in the transmission of tau from cell to cell. Oops. Oh, okay. So again, the LDL receptor seems to be involved in endocytosis of tau and its subsequent spread. So another concept is to knock down the LRP1 receptor and then possibly reduce tau uptake. And they're studying this in neuroglioma cell line to see if it can decrease the spread of tau in the brain, and again, be a potential target for the treatments of diseases involving tau spread and aggregation. So lots of promise, but one major caveat. So the idea that modulating ApoE, HSPG interactions could reproduce this potentially protective effect of the ApoE3 Christchurch mutation. Interestingly, they found 4 individuals in the PISA cohort that were heterozygous for the ApoE3 Christchurch mutation, who also had the PS1, presenilin-1 mutation. Unfortunately, all of them showed early onset mild cognitive impairment typical of the PS1 characters, which suggests that you need to be homozygous for the ApoE3 Christchurch mutation to get protection from early onset Alzheimer's disease. The other major caveat is this is a case study. It's an N equals 1, and it's now 2 years post-publication. And the only thing I could find is that heterozygotes do not protect against Alzheimer's disease. You have to be homozygous for the Christchurch mutation to get the protection, and we only have an N equals 1. So one of the concerns is that this protective mechanism may only apply to people with the PS1 mutation. And we know that the familial autosomal dominant types of Alzheimer's disease only account for about 1% of the Alzheimer's cases. So we don't know whether this would apply to sporadic Alzheimer's disease or not. Uh-oh, what happened? Uh-oh, what's going on here?
Probably can just continue, Bob. I don't think there's a problem.
I seem to have lost my presentation. Okay, well, that's at the beginning. Hold on. Okay, so here it is back again. Okay, so why isn't it advancing? Okay, so now it's advancing. Okay, so this again, this is the detour that I ended up going down. And I've been following Heiko Brock's work for quite some time now, basically because of an interest in Parkinson's disease. And believe it or not, I've been a follower of the Brock hypothesis since about 1970, which was well before Brock put forward this hypothesis. But when I was a graduate student at Vanderbilt and taking neuroanatomy, I can remember my neuroanatomy teacher telling us about the substantia nigra and Parkinson's disease. And he informed us that as soon as all the people who had survived the 1918 flu pandemic died off, then Parkinson's disease would disappear. Well, obviously he was wrong, but the fact is that there were a lot of people who did develop a Parkinson's-like syndrome from a neurotropic virus that was also circulating at the same time as the influenza virus that was causing the 1918 flu pandemic. And indeed, those people did have viral encephalitis, which led to a viral Parkinson's disease. So I've been fascinated with the idea of virus causing neurodegenerative diseases, obviously since about 1970. So when Brock came up with his hypothesis about Parkinson's disease being a viral disease, at least some of the cases of Parkinson's disease, certainly not all, as we know with MPTP and rotenone. But basically, his idea is that you can get viral Parkinson's disease either through the nasal cavity, a virus that goes in through the olfactory nerve, or through the stomach or the gastrointestinal tract, where the virus transmits up the vagus nerve, the vagal afferent, and enters the brainstem at the solitary tract nucleus, and then proceeds forward, or if it comes in from the olfactory nerve, proceeds backwards. And he's staged various stages of Parkinson's disease based on the transmission of this viral particle that is ultimately causing damage. And one of the interesting hypotheses associated with this is that this virus may have a unique kinase that is phosphorylating, in the case of Parkinson's disease, alpha-synuclein. But interestingly, he's now looking at the possibility that it's also phosphorylating tau. Okay, so this may be an exogenous kinase that's brought in by a viral infection that is phosphorylating tau and leading to the initial phases of tau phosphorylation, which ultimately leads to tau hyperphosphorylation, and then the formation of the neurofibrillary tangles. So why is this not advancing? So this is another review article that really got me totally changed from even considering the possibility that amyloid could be the initial step. And up until like 4 days ago, I was of the opinion of that, okay, beta amyloid is the gun and tau is the bullet. Now I don't think that anymore. I think if anything, tau is the gun and the bullet, and that beta amyloid is just more like collateral damage. So this review article, they talked about how the tau lesions are crucial for the pathogenesis of sporadic Alzheimer's disease, that the early pretangle disease is what happens first, and it's only later that you start see A beta deposition being present, and that it's only when there's pathological conditions associated with abnormal tau that you actually get the extracellular and aggregated A beta. And again, a potential trigger for this tau protein hyperphosphorylation, which leads to a conformational change or a misfolding of the protein. So we have now sort of a prion-like transmission of the damaged tau protein, the hyperphosphorylated tau protein that's capable of aggregating following its misfolding, is that there may be a non-endogenous pathogen, i.e. a virus, such as herpes simplex virus 1. And he's hypothesizing that it may provide an exogenous kinase capable of phosphorylating tau in addition to any endogenous kinases that could be phosphorylating tau. So obviously the hope is that these hypotheses could lead to studies that will look into the possibility of looking at new preventative and disease modifying strategies. One of the strategies that he suggested, which is very interesting, is that we start having the early stages of Alzheimer's when we're teenagers because of a herpes simplex virus infection. But it's a very, very slow process. And it takes 60 or 70 years before it actually gets to the point where you have neurodegenerative disease. So one of the possibilities he's suggesting is we should be treating people with antivirals very early. But the other obvious possibility is that maybe we should be looking at kinase inhibitors. And I'm going to start moving in that direction. But in the process of doing this, I was bouncing the idea for my abstract off of Lisa Robeson, who's one of my colleagues in neuroscience. And she said, you know what? I remember something about viral infections and beta amyloid. And she pointed me in the direction of this paper by William Eimer and Zandra Breakfield from Harvard. And basically, what they were saying is that when you get a herpes infection, and this is a mouse model, but they use some 3D human neural cultures as well, that actually the formation of the beta amyloid is a protective mechanism. So basically, the beta amyloid walls off the herpes virus and prevents it from spreading and killing you or causing neurodegenerative disease. Now, the obvious thing is that, yes, it's promoting short-term survival, but at the expense of long-term toxicity as the amyloid beta starts to aggregate and build up out of control. So more on viral infections. This is herpes simplex virus 2 infection causing an AD-like neurodegeneration in human neuroblastoma cells. Again, herpes simplex viruses are neurotropic viruses that stay there like forever. And they're suggesting that it's involved in the pathogenesis of Alzheimer's. Okay, and even going to the point of HSV2 infection causing strong accumulation of the hyperphosphorylated tau and subsequently the amyloid beta peptides of A beta and A beta 42 in their SKN-MC neuroblastoma cells. So more on herpes virus. This is a study of several different viruses. They did CMV, Epstein-Barr virus, as well as one of human herpes virus 6. And what they found was that CMV, didn't seem to be a promoter of Alzheimer's, but that Epstein-Barr virus and herpes virus 6 infections may be associated with development of Alzheimer's disease. And then here's another study, which again is solidifying the idea of herpes simplex virus being involved in Alzheimer's disease. And what they showed was that well, the first thing they said is that it may play a pathogenetic role. And interestingly, they looked at antibody titers against herpes simplex virus 1 and found that they were increased at baseline. And what they did is they took people that were having MCI and followed them for a couple of years to see if they converted to Alzheimer's disease. So they found some of them did convert, but then some of them were resistant, and they were the so-called non-converters. And interestingly, a characteristic of the non-converters was that they had higher HSV1 antibody titers, and the antibodies were had higher affinity for HSV1 than in the non-converter group. So again, this is suggesting that the herpes simplex virus is a major contributor to the onset of Alzheimer's disease. So again, this reinforces the hypothesis for a role of herpes simplex virus 1 in the pathogenesis of Alzheimer's disease. So then that brings me over to, well, what about kinase inhibitors? Could kinase inhibitors have a therapeutic benefit? And one of the first papers I stumbled across was a paper from Dr. Albensi's lab. I had originally wanted to look for Scott Turner, who is a researcher who's looking at nilotinib both for Parkinson's disease and now increasingly for Alzheimer's disease. So nilotinib is a tyrosine kinase inhibitor. These inhibs, there's a whole bunch of them. The first one was imatinib Gleevec, which was developed as a cure for chronic myelogenous leukemia. And I'll talk a little bit about that somewhere in one of the slides coming up. So anyway, this paper was showing that nilotinib, the tyrosine kinase inhibitor, was improving the bioenergetic profiling in 3xTG mouse model and astroglia, isolated astroglia from a 3xTG Alzheimer's disease mouse model. And what they found was that nilotinib improved mitochondrial function, again, suggesting that, well, microglia could be a key therapeutic target in treating Alzheimer's disease, and that the possibility that nilotinib, by inhibiting a tyrosine kinase, is preventing the phosphorylation of proteins that are ultimately leading to mitochondrial dysfunction and the death of neurons.
Hi, Brian.
So again, moving on to going back to Parkinson's disease, because again, this is something that I've been very interested in. And what this paper was suggesting was that an inhibitor of the Abelson tyrosine kinase might slow the progression of alpha-synuclein pathology and neurodegeneration, which is the hallmark of Parkinson's disease. And the reason this caught my eye is because several years ago, Jessica Wapner came and gave a talk at the NSU Honors Distinguished Speakers Series, and she was talking about the Philadelphia chromosome, which is the cause of chronic myelogenous leukemia. And it's actually a crossover of c-Abl with BCR, which generates an unregulated kinase that ultimately causes chronic myelogenous leukemia. And this was the whole, this is an incredibly phenomenal book. I recommend to everybody to read the book, The Philadelphia Chromosome. It's a really great story. And, you know, it especially promotes the efforts of Brian Drucker at Oregon Health Sciences Center in staying with Gleevec and convincing Novartis that they had to start making Gleevec available for treatment of leukemia. And it wasn't until Novartis discovered that people would have to take this for the rest of their lives, that they said, oh, well, maybe we can make some money on this. So they actually came out with Gleevec, and it's, just a wonder drug for treatment of chronic myelogenous leukemia. So again, the internalization of the misfolded alpha-synuclein, according to his hypothesis, activates c-Abl, which then phosphorylates more alpha-synuclein, and it's kind of a vicious cycle promoting the alpha-synuclein pathology within the affected neurons. And postmortem studies of Parkinson's disease patients has demonstrated increased levels of tyrosine phosphorylated alpha-synuclein consistent with the activation of c-Abl in human disease. Now, they did try nilotinib in a clinical trial, and it didn't work, unfortunately. But one of the possibilities that didn't work is that it doesn't cross the blood-brain barrier particularly well. So it may have been a case where they just didn't get enough of the tyrosine kinase inhibitor into the brain. So I think this is still a possibility. And, the fact that this author is from Inhibicase Therapeutics suggests to me that they're still pursuing this concept of using tyrosine kinase inhibitors to treat both Parkinson's disease and Alzheimer's disease. So again, more on nilotinib and another analog, bosutinib. And again, this is Scott Turner's lab out of, as well as Charlene Musa at Georgetown. And again, they're looking at Abl causing neuroinflammation in Alzheimer's disease, and trying to inhibit that with the tyrosine kinase inhibitors to facilitate amyloid clearance. That's their hypothesis, and decrease inflammation. Although again, being a Taoist, I'm thinking that they're really trying to inhibit tau phosphorylation. So what they found is that it reduced astrocyte and dendritic cell number with no effects on microglia. But astrocytes can also play a role in brain inflammation. So it's not just the microglia that cause brain inflammation. So anyway, nilotinib did decrease blood and brain cytokines and chemokines while increasing CX3-CL1. Okay, but it increased IL-10, which is an anti-inflammatory cytokine. I'm not sure about CX3-CL1. I'm naive on that one, sorry. But again, the tyrosine kinase inhibitors may regulate systemic and CNS immunity and may help treat Alzheimer's disease, where some of the effects are associated with immune hyperreactivity. So widespread distribution of tauopathy and preclinical disease. So again, this is pursuing the idea that the first step in Alzheimer's disease is abnormal tau. And the abnormal tau may be associated with the abnormal phosphorylation of the tau protein, which ultimately leads to formation of the hyperphosphorylated tau, the misfolding, and the prion-like aggregation process. And this is just showing that excessive tau accumulation in the parietal occipital cortex characterizes early onset Alzheimer's disease. So again, this is a situation where they were comparing early onset versus late onset Alzheimer's disease. They were using the AV1451 18-fluoro positron emission agent to study phospho-tau distribution. And again, saying that there was a distinct topographic distribution of tau that may influence the nature of the cognitive impairment in early onset patients. So again, this is again supportive information that tau is the initiating factor in Alzheimer's disease. So naturally, we have a study that tried to develop an inhibitor of tau aggregation. This is not a tyrosine kinase inhibitor. It's a selective inhibitor of tau protein aggregation. This was published in Lancet in 2016 to see if they could modify disease progression in patients with mild to moderate Alzheimer's disease. Unfortunately, it didn't work. So in this case, preventing the aggregation of the tau was not successful. So it's kind of back to the drawing board. But again, my thought would be we need to see if we can get a tyrosine kinase inhibitor into the brain, again, in early stage Alzheimer's. I shudder to think that we may want to be thinking about starting when people are teenagers, but certainly in the 40s or 50s, that we might want to be looking at tyrosine kinase inhibitors, and in particular, a very selective tyrosine kinase inhibitor, because we can't just go willy-nilly inhibiting all the tyrosine kinases in the brain or anywhere else in the body, or we're going to obviously interfere with the functions of life. So we've got to find some very specific tyrosine kinase inhibitors that are going to inhibit that specific tyrosine kinase. Or, you know, it's always possible it could be a serine threonine kinase that's also causing the problem. Although what I've seen mostly is the hyperphosphorylated tau, I believe is more tyrosine phosphorylated than serine threonine phosphorylated. So that's kind of a stopping point because I didn't think I would be able to get to the MAF2, and this is something that conveys cognitive resilience, and this is really fascinating. This is a paper that came out earlier this month in Science Translational Medicine. I think in the interest of time and being able to give people some time to ask me some questions, I'm going to maybe save this, and maybe I'll get invited back again. So Ben, I think I can stop here.
Okay, that's great, Bob. Thanks for that. That's lots of really interesting material. I just wanted you to know that Dr. Turner, my colleague, is actually in the audience today.
Oh, really?
Yeah, so I see him on the list.
Well, thank you, Scott. I need to come and visit you on one of my trips up to Georgetown, because I have a lab up at Georgetown.
And then I also just want to point out there is a comment from John Hardy. John Hardy, did you want to elaborate on that? I see your comment in the chat box. Did you want to unmic yourself? Unmute yourself, I should say.
Hi, yeah. Hi, thanks. I really enjoyed. Thanks, I really enjoyed that. Of course, you know, we have to explain why people whose only defect, genetic defect, is an APP gene duplication, and of course there's the other APP and presenilin mutations, go on to develop full Alzheimer's disease, whereas tau gene duplications, for example, give rise to FTD. So I think, you know, I think we do have to, you know, explain that finding. And if you cross APP mice with tau mice, the APP transgene increases the tau pathology, but the tau pathology does not alter the amyloid pathology. I think, you know, that puts amyloid, in my view, upstream of tangles.
Okay, well, you know, we have this debate about cancer. You know, cancer is not one disease. So I suspect we have a similar situation here with Alzheimer's. There's probably a lot of different variants. We already know that there are the genetic mutation Alzheimer's diseases. So yeah, I shouldn't be ruling out a pure amyloid beta Alzheimer's disease phenotype. But on the other hand, I think we have underrated the importance of tau in many cases of Alzheimer's disease.
I see another question here from Dr. Guha. Dr. Guha, would you like to unmute yourself, or do you want me to ask the question?
Yeah, I can go ahead and ask the question. First of all, fantastic talk. And so my question, like you were talking about all the different viruses like causing potential neurological problems. So like the whole world that we are suffering now, like, do you think like can coronavirus can accelerate progression or symptoms of Alzheimer's disease as well? Like, are there any reports based on that?
That's something we're studying right now. We have a grant to study this. The challenge is that ACE2, the receptor for SARS coronavirus 2, is pretty wimpy in the brain. So there's not a whole lot of target for the coronavirus, assuming it can get across the blood-brain barrier. We are thinking that the CNS problems associated with SARS-CoV-2, the so-called brain fog and long COVID, probably relate to damage to the endothelial cells in the cerebral microvessels. And basically what you're getting is mini strokes or ischemia that's causing the damage that we're seeing associated with the SARS-CoV-2.
Okay, thank you. I have another question kind of unrelated to this. Like you talked about like hyperphosphorated tau, like one of the main causal factors for AD. So what do you think, like any other modifications of tau, like be it acetylated tau, can they equally be toxic, or is it like more we are concerned about the phosphorated species of tau?
I'm gonna tell you that I honestly don't know how to answer your question. I've been focusing mostly on the phosphorylation of tau. I really haven't looked at things like truncation of the tau protein, and there's a whole lot of other things that are going on there. So I'm gonna have to punt on your question on that. Sorry about that.
No, that's fine. Thank you so much.
I see one other question in the chat box. I'm not sure who it's from exactly, but the question is, could HSV infections be a cause of presenile Alzheimer's disease, which is not genetically mediated?
I would imagine if you have a severe enough HSV infection and you have a very poor immune response to the HSV infection, that it's going to initiate the process of Alzheimer's disease earlier than it would in someone who has some reasonable defenses against the toxicity of the herpes virus.
Yeah, I asked that question. This is Naushira Pandya. I'm in the geriatrics
Hi there.
department. Hi, Dr. Speth. Fascinating. Yeah, I just wondered, because sometimes we see cases of Alzheimer's, dementia in people. Well, we don't genetically test all family members, but I'm wondering if HSV infections, in childhood or young adulthood could be severe enough to cause this. And as to the question about COVID, I know that in the clinical setting and in nursing home patients who have dementia or MCI, they're certainly more cognitively impaired if they survive a COVID infection. And right now, it doesn't seem to be reversible. They lose a lot of ground in terms of functional ability and cognitive ability once they have COVID. And I mean, your suggestions about possible etiologies are very valid. And could it also be just a general, increase in inflammation in the brain, which initially, causes delirium. And then, as we know, many people don't actually recover cognitive abilities, especially if you have an abnormal brain to begin with, once you've suffered a bout of delirium.
That's an excellent point. And, you know, one of the other things, the inflammation may affect the blood-brain barrier. And the inflammation may compromise the ability of the blood-brain barrier to keep other things out of the brain, and that those other things are what's going into the brain and causing the inflammatory damage to the brain.
Thank you.
Richard.
Yeah, Bob, great. Lots of ideas come out of your presentation. You know, last month I talked about the NLRP3 inflammasome, and of course the activation of that inflammasome by viruses is really a very primary role that it plays. So whether it's HSV that we're thinking about or COVID, both of them, you know, could be stimuli that promote the NLRP3, which is activated in Alzheimer's disease and leads to cell death, you know, by that pyroptosis mechanism.
Absolutely. Yeah, it seems like just about every disease these days has an inflammation component associated with it.
Well, Bob, it is 1 o'clock. I want to thank you for giving such a great presentation today. We had a really nice turnout. We will have another presentation next month, of course, in December. That'll be the last one for the year. And if you joined late, or if your friends or colleagues could not join us today, there is a recording link that will be made available on my social media. So on LinkedIn, Twitter, and Facebook later today, if you look at my social media, so you can follow up and ask Dr. Speth additional questions and rewatch the video or watch it for the first time. So again, Dr. Speth, thank you for that great presentation, and happy Thanksgiving to everyone. I should have said that at the beginning, but thank you for coming today.
Thank you for the invitation.
Okay, thank you, everyone. Bye-bye.
Bye.